I'd like to start just by having a look at this disease, which is possibly more common than a lot of people think. Can you talk a little bit about what it is these patients experience and what their current options are, and how we might think about new ways of looking at the disease?
Well, the target unmet medical need that we are approaching here is called transthyretin amyloidosis. Amyloidosis is a group of serious diseases which involve misfolding of proteins, the body's own proteins, which are normally soluble and easily metabolized and broken down. But when they misfold, they can sometimes form insoluble fibers which accumulate in the tissues, and which, for reasons which are still not well understood, the body finds it very hard to clear this material away from the extracellular space between the cells. And so the protein fibrils, which are called amyloid fibrils, increase and accumulate, forming deposits, amyloid deposits, which disrupt the structure and therefore the function of whatever tissue or organ they are in. And eventually they damage the function so much that patients become clinically ill and eventually die. So it's a universally fatal disease if it isn't adequately treated. It was discovered many years ago that wild-type normal transthyretin without any mutation can also make amyloid. And this is usually found at autopsy in elderly people with increasing frequency as people die at older ages, and was always considered to be an incidental finding without any great pathological or clinical significance. However, very occasionally people would be found with no transthyretin mutation who developed amyloid in their heart of this type and went on to get heart failure and to die. We used to see in our center, the National Amyloidosis Center, which is the largest, most diverse population of amyloidosis patients in the world, we used to see and diagnose maybe 1 or 2 such patients a year amongst 5,000 or more attendances at the center.
Right.
So very rare. In the last few years, due to advanced imaging and new developments and understanding of how to diagnose the disease safely and non-invasively, it turns out that something like 3 to 5 or even more % of men over the age of 70 have got or will develop this type of wild-type transthyretin amyloidosis. So far from being rare, it's an extremely prevalent cause of heart failure and heart rhythm disturbances, which is inevitably fatal. Now there are 2 forms of treatment that have lately emerged.
Yeah.
The first one is so-called stabilization treatment. So a very innovative scientist called Jeff Kelly in the US had the idea of creating small molecules that would be bound by transthyretin and would stabilize it and stop it misfolding and aggregating as amyloid fibrils. And that ended up in the production of a drug which is now prescribed widely around the world, not actually in the NHS in the UK, because the regulators haven't approved it for that purpose, but it's used very widely elsewhere in the world as a stabilizer of transthyretin. And the clinical evidence is that it somehow slows the progression of amyloid deposition, and that's obviously clinically beneficial. The other part of the story is that all the work up to that point, looking at the efficacy of stabilizers of transthyretin, preventing them from making amyloid, all of that was based on what turns out to be an artifactual model of amyloidogenesis. So Jeff Kelly's great, very innovative idea of stabilization was spot on, but the model he tested it in turns out not to be physiological. What he did in order to make transthyretin into amyloid was to denature, basically wreck the transthyretin molecule by incubating it in an acid condition for 3 days. And there is no way that condition can exist in the human body or any other living organism, which could be relevant to amyloid deposition. It's an artifact. And when you actually look at what happens to transthyretin when it's been wrecked in that way, you've got great aggregates of protein, but you haven't got more than a tiny number of actual amyloid fibrils. Amyloid, if it's there at all, is very sparse. So now we come on to what actually is the process by which transthyretin makes amyloid. And it turns out to be much more complicated than just denaturing it, wrecking it under abnormal conditions. It turns out that this requires 2 things to happen. First of all, it needs a cleavage, a clip in part of the transthyretin molecule, a very specific break in the amino acid chain which constitutes the whole protein, and it needs to have mechanical forces applied to the protein. The mechanical forces shake up the protein a bit and slightly destabilize it, which doesn't actually do it any harm per se. But if there's an enzyme around that can make this specific cut in the protein, the mechanical forces facilitate that happening. And as soon as that cut has happened, there's a fragment released from the whole transthyretin molecule, which catalyzes, encourages the rest of the molecule to misfold and to aggregate as amyloid. And that process, which we call the mechanoenzymatic mechanism of transthyretin amyloid fibrillogenesis, that process is A, extremely efficient, B, it happens under completely physiological conditions, such as would exist in the tissue spaces where amyloid forms in the body, and C, it yields large amounts of totally authentic, genuine amyloid fibrils. It doesn't leave just clunks of junk.
Yeah.
It produces the real deal amyloid fibrils. Now we go back to the stabilization molecules. Okay, when we start to compare the capacity of our bivalent superstabilizers with tafamidis and other molecules of that type, the super potency is even more radically present. And we have now worked out at a detailed molecular level exactly why that is the case. And the answer lies in the fact that transthyretin is a symmetrical molecule which has two binding pockets, each of which can bind thyroxine. But it's been known for ages that these two binding pockets have what's called negative cooperativity. A positive cooperativity is if doing one thing helps you to do the next thing. Negative is just the opposite. When transthyretin binds one thyroxine or stabilizer molecule in one pocket, it's very hard for it to bind the other one. The affinity is radically reduced for that binding. So there's a high affinity.
So presumably that would be the same for tafamidis if it was binding into one of those pockets.
Yeah, once tafamidis is in one pocket, the transthyretin cannot bind the second tafamidis in its other binding pocket unless there is a very high concentration of the drug available. And you can't achieve that desirable high concentration in the body in clinical conditions with the doses that are possible to give and so on. Just isn't possible. And that applies to all these stabilizers which only occupy a single pocket, what we call monovalent stabilizers, and our ones we call bivalent stabilizers. And a very important point about our bivalent stabilizers is that not only do they simultaneously occupy both binding pockets, they also occupy a third binding pocket, which is the channel down the middle of the transthyretin molecule. Because what these bivalent stabilizers very cleverly do is they insert themselves into the first binding pocket and then they thread themselves down the middle of the transthyretin molecule where there's a very narrow pore, but these little molecules go all the way through and they plug up both binding pockets and they occupy the channel in between. And so we believe that there is an enormous possibility, provided, as I said at the outset, we can turn one of these molecules into something which is safe and well tolerated in patients, provided we can achieve that. This approach will completely arrest new transthyretin amyloid formation.
And presumably that gives the body an opportunity to actually clear the rest of the amyloid away.
Yes, it will give it, absolutely. Once you stop amyloid accumulating, we now know from the gene knockdown studies that transthyretin amyloid can be reabsorbed and cleared away with clinical benefit. It takes time, it takes months, but it can happen. So that would be a very good thing.
So will we see now a pill simply that somebody would take, is that something that you think is possible?
Yes, I think it's absolutely possible. I think that the risk in this approach, the only risk, is that we won't succeed in making something which can be a medicine. That is always a very dicey process. Anybody who knows about drug development, you've got to cross lots of hurdles, scientific, clinical, regulatory, and so on. But provided we can cross those hurdles, and we have very promising molecules, which are looking so far, the preclinical pharmacology, which the Tech Fund has so generously funded for us and supported, that is all looking very promising. We're close to candidate selection and so on. So the risk will only be in those aspects of drug development. Scientifically and clinically, if we have a safe, well-tolerated molecule, it will work therapeutically. That's 100. There's no risk there. That is 100% guaranteed. I mean, short of some completely unprecedented phenomenon, and anything can happen when you put a new chemical entity into a person. But you know, that the scientific basis of it is irrevocably strong, and it will, you know, it will work. So it will arrest amyloid, new amyloid formation, and crucially, it will be an approach to preventing amyloid formation. There are the 10,000 people in the world who have families, you know, half the children of these 10,000 people, half their children will have the bad gene and will be at risk of getting that type of amyloid as they get older. Why should they ever get it? We'll give them their pill. They'll take it once or twice a day. They will never get the disease. And likewise with people aging. Now we're talking on a much more massive scale of intervention. If this is a safe medicine, you would take it like a statin, you know, from age 60 or something. You know, you would take it and nobody would get wild type cardiac amyloidosis again. That is the you know, the desirable outcome which we could achieve if we can produce a satisfactory medicine. So my final point I'd like to make about all of this is that you will have gathered from my descriptions of all the different ways in which we've approached the problem that it is a truly multidisciplinary affair.
Absolutely.
And you know, this has always been the way that we've worked in my lab, but this has been the sort of the acme or the apogee of it, in the sense that we've, you know, tremendous collaborations with biophysicists, with NMR and X-ray crystallography, and experimental animal models, and the clinical, you know, basis of it all from the National Amyloidosis Center, and knowing what happens in the patients and what the other treatments are. It is a truly multidisciplinary effort, which has produced a synergy, you know, way beyond just the sum of the parts. It's multiplied our capacity to make progress and understand, and all of it has only been made possible by your generosity, Simon.